All right, good evening, students. Welcome you back. I hope you are enjoying the sessions which we are giving you. Our purpose is to give you maximum information and to give you and solve questions which are important for the exam students. Our time is short that we know, so we have to cover the syllabus which is very important. And last time, a small recap is given to you that we are talking of amino acids, and the important point to note is that students, what we were talking mainly, we said that on it was found that proteins on hydrolysis or proteins on hydrolysis produced alpha amino acids mainly. This is an important point because we are coming to protein students. Since our we are going to discuss student proteins. And what we are saying, student proteins, what are protein students? They are nothing but alpha amino acids. Proteins, we are going to discuss proteins. Student proteins, you are important, very important for you. You are eating every day and you need proteins. Each one of us needs protein, students. And it was found that on proteins were hydrolyzed, students, hydrolyzed. And you have seen earlier also in saccharides, when I do the hydrolysis, what I was getting. Polysaccharides were giving monosaccharides. So we were getting monosaccharides, which helped us in saying that this is the structure of the disaccharide, sucrose, maltose. All this we discussed with you. What are the glucose units which make up these molecules? Most of the times you are using glucose, students. Glucose molecules are being produced. They are combining at C1, C4 or at different combinations, alpha D glucose, beta D glucose. Like this, you are able to get different type of carbohydrate, students. Now here we are saying when proteins are hydrolyzed, they found that only alpha amino acids are obtained. Alpha amino acid, I said the best way to, what we said, this is a carboxylic group, student. The next carbon, this is called alpha carbon. This is my beta carbon. This is called gamma. This carbon is called delta, like this. So the NH2 group is where? At alpha position, students? At alpha position. So if you have this structure, student, you can see what I am saying. This is a COH group and a carbon next to the COH group, there is a NH2 group. Now here you can have different groups, as you can see. And you can see if I write student H and this I write R, student. R means this I will vary, this I will change and I will get different amino acids. Different amino acids will be obtained, students. So these amino acids can contain other functional groups also, students. But alpha amino acids means COH group, adjacent carbon has got NH2 group. So the best way to, and further it was found, student, that they belong to L series. Now this is the important point, students. I told you about two series in glucose also, D series and L series. D and L, capital D and L, we told you they were derived from glyceraldehyde.
Yes, sir.
And when the OH group was represented on the right hand side, we said it is D, and OH group on the left hand side, we said L. And when I kept on writing D glucose, what it meant to you? When again and again we were writing D glucose. You have seen D plus glucose or D minus glucose. And in this, we said that the lowest asymmetric carbon, the chiral carbon, carbon which is optically active, which has four different groups attached to that, student.
Yes, sir.
Lowest asymmetric carbon was carbon 5 in glucose. That had OH group on the right hand side. That is the D glucose. If I take the mirror image of that, then it will become L. That will be a stereoisomer, which will be an enantiomer of D glucose. These are very specific things because their biological activity changes, students. When you buy a bottle, you will see D glucose written, not L glucose. Why? Because these are biological specific compound students inside your body. This is very important point. As you will study in biology, you will see more in biochemistry, you will study. And it is found that these amino acids belong to L series. So what we do, students, we we are representing like this now, an easiest way. This means NH2 group. Now we have NH2 group alpha position. This is my alpha. This is a carbon. This is a carbon alpha position. ठीक है? This is my carbon student. Alpha carbon. Now NH2 I am writing on the left side. L. If I write on the right side, it will become D. But it is found that they were belonging to L series. L series. Now we said this to write different amino acids, we put a group R student. This I will change student and I will get different amino acids. And this is the way to, I am talking of natural amino acid students. Natural amino acid. Okay? Clear?
Yes, sir.
And there are 20 natural amino acids which are listed in the book student. Most common one, 20 natural amino acids. And this book, in the book you will see they have given a list of 20 natural amino acids. This is always there in them. R is changing. R is changing. If I write R equal to H, then that is called glycine, students. What is the formula of glycine? We told you. NH2 COH. I am writing like this, student. This is my H and this is my H. This is glycine. It is actually sweet in smell, so the name was given glycine. Their names, you see that they have very difficult, different type of names. There is no scheme of naming IUPAC or anything. And they can be represented by 3 letters or 1 letter. As you will see, GLY glycine, alanine, valine, leucine, and G, A, capital V, and L. Like these symbols have been used for them. And list includes 20 amino acids in the book. You can go through them. And interesting point was, students, what was the interesting point in this, student? That in the table you will see 2 types of amino acids. Now a question has been asked on that. Where a question is there, I will always be giving you. Question was in the paper, what is the difference between essential and non-essential amino acids? You have 30 seconds to answer this question, students. What
Yes, sir.
is your answer? You will get 1 mark, 1 mark for this answer. See how the questions are put. Very simple question, but this is the important point that we can easily get answers, full marks in this chapter. So we have to learn it, students. What is the difference between essential and non-essential? Essential means what?
Sir, essential are the ones which we get from a diet,
From
from outside, and non-essential we get from diet.
non-essential.
Yes, sir.
Now some mistake is there. The meaning itself is there. Non-essential means they can be made by the body itself.
Body,
students.
yes, sir. Yeah, and essential from outside.
So they can be synthesized. They are synthesized within the body, students. Synthesized. What I'm saying, they are synthesized, student, by the body itself, within the body. Your cells are working, they can make them. So when if the body can make them, synthesize them, we don't need it from outside. But if body cannot make them, then what will happen? That is why they are called essential, because their deficiency will cause a lot of problems. A lot of diseases, students, if deficiency is there. Because your proteins will also be acting as enzymes in your body. What enzymes are? Basically, they are also globular proteins. Now the story is clear. They will give example of one and one. I said those, it is written here. These asterisk where there is a star, you see, they are essential amino acids. Lysine, arginine, isoleucine. Out of this is a star, this is a star, this is a star, student. So they are essential for us we have to take it in our diet, student. And those which have not starved, they are being manufactured within the body. So when body can make it, then we don't need it. Got the difference, student?
Yes, sir.
I mean, what we have to write, we should be very careful. We are doing now biochemistry, biomolecule students. Okay. So we have seen the L series, how to draw the structure, students. Only one alpha amino acid, which I have written natural, is not optically active. Name that, because question can be put like this. Is not optically active. Which one is that, student? Question 2. Which alpha amino acid is optically inactive? What is your answer? There is no chiral carbon. There is no carbon with 4 different.
Sir, glycine.
Glycine is the correct answer. So we can draw the structure of glycine. COON. NH2 on the left hand side. L series. This is my H. This is my H. Okay, the other two groups are same. So there is no chiral carbon. It is a chiral molecule. A chiral molecule. We have learned what is optical activity, student. It is, this carbon is not asymmetric. This carbon is not attached to 4 different groups. Therefore, this is optically inactive. Wherever something is different, student, that can become a question. So be careful. Okay. Now the classification of amino acid was done on what basis, student? We told you that amino acids have got basic group as well as acidic group. Which is a basic group in this? Basic group is NH2. It has a lone pair of electron. It is a Lewis base. And acidic group is, student, COH. Therefore, there can be more NH2 or more COH group in the different amino acids. I'm not saying there is only one NH2 or one COH group. If they have equal number, then they will be neutral. If more NH2 group, they will become basic. If more COH group, what will happen? They will become acidic. So they can be neutral, acidic, or basic. And important point to note is that, students, because what you have here, if I write something like RCHNH2 C double bond OOH. This is our acidic group. This is our basic group. Yes or no? Tell me.
Yes, sir.
There is a lone pair of electrons. So what can happen? This proton can come here. There can be, this proton can go and attach with NH2, students. So what will this result in, student? What will you get then? NH3, if the hydrogen will come here, the nitrogen will get 4 bonds, then nitrogen will become plus. Nitrogen will become plus, student. This was NH3 plus now. And what is left here? CO minus. So there is a positive and negative charge created in the amine, student. There is what we say dipolar. This is called zeta ion. The question was asked, student, in the paper, what is a zeta ion? Question, what is zeta ion? This is called zeta ion. There is a positive and negative charge which is present in the amino acid. Okay?
Yes, sir.
One more question. What is a zeta ion? See, simple questions. We have to draw it. So except glycine, all natural amino acids are optically active, since alpha carbon is asymmetric, and most naturally occurring amino acid have L configuration. We told you very particularly. So this is then represented by writing NH2 group on the left hand side. Now the question is, students, when I said proteins will be the result of the when amino acids will combine, student. And you will find when more than 100 amino acids, combine, students, then what will happen? The molecular mass becomes more than 10,000 U. 10,000 U student, then that is what we call as proteins. So proteins will be formed, student. What are proteins then, student?
Yes,
Proteins
sir.
means that there are more than 100 units of amino acids. They will join, students. They will join with each other. And then what will happen? More than 100 amino acid, the molecular mass will become, student, more than 10,000 U. So this is what we mean by a protein. This is what we mean by protein, students. Now the question is how they will join. That is important point, students. And what we told you, students, what we told you, very important point, that what type of bond is there in them. As we said that in these compounds, student, if I write like this is my amino, this is my NH2 group, student. There is a COOH group also.
Yes, sir.
At alpha position, this is my alpha position. And there are other molecules. So if I write something like CH2, C double bond OH, student, and NH2, this will continue. I mean, such. What will happen, students? This here is what they will combine, student. This H and OH is lost. What will happen? If H and OH is, these two amino acids are joining through which bond, student? What happens now? You have COOH, CH2, this is NH, this bond will become CO, and CH2, NH2. They will further keep on combining like this. Now what is this bond called, student? This is CONH. What is this bond called, students? This is called a peptide bond, student.
Okay, sir.
The question is asked in the paper, what is a peptide bond? Student, what is a peptide bond? Is a question in the paper. Has been asked many times. In the monosaccharides which were combining to form di or polysaccharides, what type of linkage was there, student? What is that known as? Glycosidic bond.
Yes, sir.
Now I am talking of these amino acids when they are joined more than 100 units through peptide bond, student, then we get proteins. So this is an important question then for the exam. What is a peptide bond, students? Has been asked in the paper. Therefore, you can say when two amino acid units will combine one COH group of one and one NH2 group of other with the loss of water molecules, they will form
Yes,
a
sir.
bond CONH, that is called peptide bond. What the definition? So amino acids are linked how, student? Peptide bonds. Any doubt? This question will be asked in the paper. What is a peptide bond? You can make your definition now. We have shown it. This is another way you see that. This is CH
Yes,
group
sir.
NH2. The two are minus water. This is called peptide linkage or peptide bond. So the different amino acids will be linked together through a peptide bond, student. If there are two amino acid linking, that means two amino acid mean dipeptide. So two amino acids are linked, student, and they are called dipeptide. Three amino acids will link, tripeptide. How many peptide bonds will be there? How many peptide bonds will be there, students? So you will see that the number of peptide bonds If two amino acids are linking, one peptide bond is formed. If three amino acids are linking, two peptide bonds are formed. If four amino acids are linking, then how many peptide bonds? Three. Clear?
Yes, sir.
Okay. So you are now having an idea, student, that let me write here itself. What we are saying, student, that the amino acids can link together, students, and if two amino acids are linking, if two amino acids are linked, then what we call it dipeptide. If three amino acids are linked, student, what I will call it tri tripeptide.
Yes, sir.
And if four tetra, five pentapeptide, six hexa, and more than 10, when more than 10 amino acids are linked, we call it polypeptide. ठीक है? Polypeptide.
Yes, sir.
So students, polypeptide, protein is a polypeptide then. Protein is a polypeptide. And when more than 100 units are linked through peptide bond, you get proteins. You get proteins. So proteins, you see, is a quite a complex molecule, high molecular mass molecule, high
Yes, sir.
molecular mass molecule, and it is made from amino acids. And there are certain students conformations which also have less than 100 units, but are well defined conformations like proteins. They are also called proteins. For example, insulin student. Insulin is also a protein student. Actually, it has only 51 amino acids. There are only 51 amino acids which are present in insulin. This is also
Yes, sir.
put in the category of proteins. Okay? So what is the definition of protein? Question is, what are proteins? We can answer. When more than 100 units of amino acid are linked through polypeptide bond, students, with molecular mass more than 10,000 U, that is our protein. From chemistry definition, student, when we are going chemistry paper, our answer is biochemistry paper. So chemistry, from chemistry point of view. Okay? You will write that proteins I am eating and they are essential for the body. That is not the answer for the chemistry paper. Got it?
Yes, sir.
All right. Now, one student told me, sir, I will write this definition. I am eating protein every day. I said, I am talking of chemistry paper. I am not talking of GK paper. Why? And biomolecules you are studying in chemistry. In bio, you can do that thing, not in chemistry. Okay. Here are mainly our chemical compounds we are talking about. Now, the proteins classified in two types based on their molecular shape. Every time we talk of classification in chemistry, student, what classification is there for proteins?
Hmm.
You are saying you are knowing more than me in proteins. If you have biochemistry student with you in biology, fibrous protein student. What is a fiber? Fiber thread type. Fiber made from what? Fibers thread type. See? How you will get thread student when polypeptide chains? You are saying there are amino acids, they are linked. Okay? They are linked. How many? You are saying more than 100 and you are getting proteins. And always what is there in between them? Peptide bond. What peptide bond? CONH. Okay? Within it, if there are two amino acids, how many peptide bond will be there? One. Three, two, four, three. Once they ask what is the number of peptide bonds when the amino acids are 10. How many peptide bonds will be there if the amino acids are 10? 9. So be careful about the questions.
Yes, sir.
So fibrous proteins are polypeptide chains. They run parallel and are held together by hydrogen and sulfide bonds. So fiber-like structure is formed. They have fiber-like structure. And these are fiber type structures. There are chains, student, chains of amino acids. ठीक है? And these chains
Yes,
are
sir.
held by, chains are held by, there are hydrogen and sulfide, disulfide bonds. Hydrogen and disulfide bonds, students, in them. This is a point to be noted. They will ask you in the paper what type of bonds are there in the fiber-like structure. Then what is your answer? What type of bonds are there in the fiber-like structure, student? What is your answer?
Yes, sir.
Hydrogen and disulfide bonds. This question has been asked that in the fibrous proteins, the chains are held by which atom? Hydrogen. What type of bonds are there, students? Hydrogen and disulfide bonds. These are insoluble in water. Insoluble in water, students. Any example? Any example of
In,
fibrous protein? Do you know any example of fibrous protein, student?
so let me think.
They are present in hair, wool, and silk.
Oh, yes, sir. Yes, fine. Fine.
You have heard about keratin.
Yes, sir. I've heard of keratin.
You see, Myosin is also a myosin is the name of a protein, student, which is that present in the muscles. This is these are examples of fibrous protein, student. We have to learn such thing because they can ask you give a name of a fibrous protein. At least one or two should be known to us. Keratin and myosin. They are fibrous protein, students. Second one are globular protein, students. So this is the differences in the structure. This was fiber like. When the chains will fold, student, and coil around, what will happen? There are chains of amino acids. You have chains of what, student? Amino acids. And if these chains, student, coil around, student, what will happen? They will give a spherical shape. That was what shape? Fiber type of shape. Daga.
Yes, sir.
This is spherical shape. So these are usually soluble in water. Sometimes they ask a difference, student difference. They are soluble in water. These are soluble in water. And the examples are insulin and albumins. Insulin and albumins are examples of globular protein, student. Okay? At least
Yes,
one or
sir.
two names should be known to us if they ask in the paper. You have noted down, then I will proceed further, or you will note down, students, everything, because
So copy in 2 minutes.
these points can be asked in the paper. One question, 1 mark can be there.
Yes, sir. Copied.
Okay. Now the more important questions are coming: structure and shape of the proteins. Structure and shape of the proteins, students. These important questions are given on structures. Usually protein structure is three-dimensional arrangement of atoms in an amino acid chain molecule. What is it? Amino acid only?
Yes, sir.
And we have only shown few examples of amino acids. Interested students can go and see a long list of amino acids. They contain other groups also beside NH2 or COH. But the alpha amino acid we mentioned particularly because on they all give alpha amino acid on hydrolysis, which they give which amino acid? Alpha amino acid. Alpha amino acid become important for paper. Alpha amino acid, and they are polymers, basically polypeptides. We define what is a polypeptide. What is a dipeptide? When two units of amino acids join by a peptide bond. When more than 100 join, that is called polypeptide. ठीक है? So there is a sequence of amino
Yes,
acids.
sir.
We have listed 20 now. They can form a sequence in different ways. Same amino acid or different amino acids. So many possibilities are there. 20 amino acids can join in many ways. Sequence. So by convention, a chain under 30 amino acids is often identified as peptide. If there are less than 30 amino acids, we say it is a peptide. Protein, we are saying then more than this amino acids are there. So their structure has been defined in primary, secondary, and tertiary form. First, I am saying primary structure of protein. What you mean by primary structure?
Hmm.
We are saying that amino acids join. The sequence of amino acids, student, will form the primary structure. And how many types of proteins are there? Large. It is not that only one protein is there. You know, from different food stuff, you are getting different type of proteins. They are essential for the body. Why? Because the sequence of amino acid will decide what type of protein is this. That primary structure means, student, the sequence of amino acids. By
Yes, sir.
out of 20, which one are joining? If different, I can keep on changing, then the structure will change. Sequence will change, the amino acid, the protein will change. So the first thing I'm saying, primary structure is defined by the sequence of amino acid. In other words, student, different sequence of amino acids will give rise to different proteins. ठीक है?
Yes, sir.
Clear? Because polypeptide is formed by how? Polypeptides are formed by the amino acids. Now, out of 20, which amino acids are combining? In one case, you are saying, sir, leucine and isoleucine. In other cases, some other. And so many sequences are possible. So different sequence will give different protein. And the sequence is deciding the primary structure of protein. He will ask what is the primary structure of protein, then what is our answer? It is decided by the sequence of amino acids which are forming the polypeptides. So there can be one or more polypeptide chains in the proteins. Each polypeptide in protein has amino acid linked with each other in a specific sequence. So there is a specific sequence, definite sequence, ठीक है?
Yes, sir.
Which will give rise to a definite protein. And if the sequence will change, then protein will change. This is important point because when proteins are destroyed, students, on heating or by change of pH, the primary structure is not destroyed. The sequence of amino acid is not destroyed. Only outside structure is destroyed. Second, I am coming to secondary structure of protein, student. The secondary structure means the shape in which long polypeptide chains can exist. Now we are talking of the shape of these polypeptide chains. Polypeptide chain is formed by a definite sequence. Okay? Now the shape is something what shape they take, student. And it has been found experimentally, they are existing in two different type of structures. What type of shapes they are found to exist in? Two different type of structures, students. One is called alpha helix, and other is called beta pleated sheet structure. Beta pleated sheet structure. There are two types of shapes, students, they take. These polypeptide chain, students, they take two different shapes. Therefore, they have two different structures. This is called the secondary structure of proteins. Why they arise, student? Why such shape they take? Why will anything take a shape, student? There must be some bonds formed, student.
Yes, sir. Obviously.
And here, what type of groups you are having? You already know in this what type of group you have. You have CO and NH type of group, students. And what will happen, students, then? If they all take a shape or they will fold themselves, there will be a regular folding of polypeptide chain due to hydrogen bonding, student. This nitrogen and oxygen, you see, nitrogen is electronegative, positive. This is negative. So what can happen? Formation of hydrogen bonding. And if hydrogen bonding will be formed, student, they will start shaping. They will take a shape, student. Some bond is there, na hydrogen bond, student, attractive force. Because there is a group CO and H, student. So there is hydrogen bond formation which can take place within the peptide bonds. Okay? You have noted,
Yes,
then I
sir.
will proceed further.
Copied.
Copied or copying?
Yes, sir. So copied. I'm doing simultaneously.
Okay, do it.
Yes, sir.
Oh,
Proceed,
okay.
please.
So here we have shown alpha helix is a right-handed coil strand. This is our alpha helix. You can see coiled strand. The side chain substituents of amino acid group in an alpha extend to the outside. Hydrogen bonds form between the oxygen of each CO and in the strand, and hydrogen bond of each NH group for amino acid below it in the helix. It is a helix type of structure, student. See, it has called, and you can see here we have shown this is hydrogen, this is oxygen. Within the molecule, student, hydrogen bonding is taking place. And because of this, student, these hydrogen bonds make the structure specially stable. This becomes stable, students. As you can see, what is happening within the molecule, there is a hydrogen bonds. Hydrogen bonds are formed. So within the molecule, you had CO group and NH. Both these groups are present. Okay?
Yes, sir.
And what will they do? They will form hydrogen bonds. And the side chain substituents of the amino acid fit in beside the NH groups, beside the NH group. Because there are amide bond, students, CO, NH, as you can see, this is CO, this is the hydrogen coming, and CO and NH hydrogen. So they coil, student, coil strand, which is called alpha helix structure. Alpha helix. What is this called? Alpha helix structure. So one
Yes, sir.
possible structure is this one. The shape they can take. They will ask you why this shape exists due to what type of bond, student. What you are saying? There are stable hydrogen bonds between CO and NH groups. It becomes a coiled strand. In the nature, it happens by itself. We are not doing anything. But bonds have to be, we have found what bonds are there, student. That is important point. The structure has been determined. It's unbelievable. Can't you see that? Okay, if you will say yes, I will proceed further. You can note down each and
Yes,
every
sir.
point. Each and every point is important. They can become a question in the paper, students.
Yes, sir. Yes, sir.
They won't ask you to draw this diagram. They are given in the books. They are only to show the formation of hydrogen bonds. This hydrogen bond is formed within the molecule, within the polypeptide chains. Can I proceed further, students?
Yes, sir. Yes, sir.
Okay. Now you see the second one which we told you is beta sheet. Now here what is happening, students? The hydrogen bonding is taking place not within the molecule, but between different strands. So here what we are having intermolecular hydrogen bonding. Intermolecular hydrogen bonds are formed. That coiled because the hydrogen bonding was within the molecule, intramolecular hydrogen bonding between the CO and H group. Now here what happens, you have chains now, you have polypeptide chains, and within them between the intermolecular hydrogen bonding. So hydrogen bonding in beta sheet is between strands rather than within the strands. That was intramolecular hydrogen bonding. This is intermolecular hydrogen bonding. Therefore, they form sheets which are lying side by side. The carbon and oxygen in one strand, ठीक है, bonds with amino hydrogens of adjacent strands. अरे you have different strands, students. ठीक है? The strands will join with each other through hydrogen bonds. So what do we call that? Intermolecular hydrogen bonds. And that is why they call it sheet type. Sheet type. You can see this is one sheet. This is
Yes,
another
sir.
sheet.
Hmm.
They are joining by, you can see the hydrogen bond between the sheet, students. So they are joining by hydrogen sheet, student. So this becomes beta pleated. The structure extends. The two strands can be either parallel or anti-parallel depending on whether the strand direction and terminus or C are the same or opposite. The anti-parallel beta sheet is more stable due to more well-aligned hydrogen bond. So hydrogen bond is still happening, but it is happening between the different sheets, student, different strands. That was within the same strand. There was intramolecular hydrogen bonding. This is intermolecular hydrogen bonding. So two
Yes,
types I
sir.
told you, beta sheet and alpha helix structure. This is the secondary structure of protein. Okay.
Yes, sir.
All right. We are proceeding further, student. Then we are coming to the tertiary structure of protein, student. Primary, secondary, and tertiary. The tertiary structure of protein represent overall folding of the polypeptide chains. Further folding of secondary structure takes place, student. Secondary structure has been formed by hydrogen bonds. Further folding takes place of the secondary structure and it gives rise to two major molecular shapes. Further folding gives rise to two molecular shapes, fibrous or
Yes, sir.
globular. Two shapes are formed now, fibrous or globular, and which we define that proteins are two types, fibrous and globular. Now, there should be some forces to stabilize. The question was asked, what are the main forces? What are the main forces, students? ठीक है? Which
Yes,
stabilize
sir.
2-degree and 3-degree structures of proteins. Structures of proteins, student.
Yes, sir. Mm.
Our answer is there are hydrogen bonds, disulfide linkages, van der Waals forces, and electrostatic forces of attraction. Which will
Yes,
be
sir.
hydrogen bond, disulfide linkages, van der Waals, and electrostatic. Electrostatic means there are some ions. You have seen formation of positive and negative ions. Dipolar ion I have shown you in the amino acids. Because NH2 group is basic and COH group is acidic. So what type
Yes,
of
sir.
forces are there, students? Important question. This question has been asked. What is our answer? What is our answer? Tell me. Which bonds? Hydrogen,
Um, yes, sir. Van der Waals and
disulfide,
disulfide linkages.
and
Yes, sir.
electrostatic
Static forces of attraction.
attractions. This is all we have to write. 1 mark questions are given. The overall three-dimensional shape of protein molecule is tertiary structure. So tertiary is
Yes,
the
sir.
overall shape of the final protein molecule. The protein molecule will bend and twist in such a way to achieve maximum stability or lowest energy states. You, why all this is happening, student? Why all this is happening? As I said, the molecules will take shape, students, which is the most stable shape. As in told you in every theory that things are going to be more stable, students. Molecules
Yes,
want
sir.
to achieve more stability. So they will take a shape which is most stable or which has the lowest energy, student. That is the point. You know,
Yes, sir.
lowest energy states are the most stable. Anything which has high energy is not stable because it will react. Things want to be stable, student, stable. And molecule will take a shape, student, on the basis of attractive or repulsive forces which are present in it to become more and more stable.
Yes, sir.
In chemical bonding chapter, that's what we said. There are electrical electrons, students, in them. They are repulsions and attraction plus minus charges. All this is there. So they have to balance the forces so that they acquire the lowest energy states. They become more and more stable, students. So three-dimensional protein may seem irregular and random. It is fashioned by stabilizing forces due to bonding interactions between side chain groups of amino acids. Because there are
Yes, sir.
side chains also of the amino acids, so what will happen? They'll try to coil around, they will try to associate through hydrogen bonds or any other linkages, which we are saying disulfide linkages. There are plus minus charges, electrostatic, so they will take shape, student, which is the most stable form and lowest energy state. That's why you are getting these shapes. Anything, why it should be shaped anything. Tomorrow somebody says why the earth is
Hmm.
spherical, why it is not rectangular, students.
Nice.
Because if it is rectangular, it will not be stable, then it will dissociate into a more stable state. More stable shape it will take. So whatever more stable state it could take, it formed.
Yes,
Otherwise,
sir.
it would be dissociating from unstable to stable states. Is it?
Hmm.
That is the sense we are talking about everywhere. Stability, lowest energy state.
Yes, sir.
And that is decided because there are atoms, student, which can form bonds, student, bonds, and forces of attractive and repulsive forces are operating. So there is a balance of these, balance of all these. So they take three-dimensional shapes, which was talking about in the proteins. Globular,
Yes, sir.
you can see in two shapes which we gave you, fibrous and globular. Maximum stability. Now, quaternary structure of protein. Some of the proteins are, what do you mean by quaternary structure of the protein? This is the last thing in the protein which we are talking about. Up to now, I said they are made up polypeptide chains, but there is a
Yes, sir.
possibility, student, there can be more than one type of polypeptide chains. What will happen then? One polypeptide chain is formed by one sequence of amino acids. If there is another polypeptide chain, everything is possible.
Hmm.
So what will happen? There inside this there are subunits. So then
Yes, sir.
these arrangement of subunits with respect to each other is known as quaternary structure, students. This is called quat. Here you can see there are two color different.
Okay.
So they are two different polypeptide chains. They are two different units, student. They are
Polypeptide.
called
Yes, sir.
subunits.
Okay.
So in this case, what you find, they are composed of two or more polypeptide chains. Everything is of everything is coming from polypeptide chains. But if there are
Yes,
different
sir.
polypeptide chains, different colors we have shown, then what will happen? They form a structure which is known as quaternary structure.
Okay, sir.
So this is example of the quaternary structure. We don't, I'm not going to very big details here because that is not required for our course. Those students who are biology, they will study in that.
Yes, sir. I did.
So our course finishes up to that point. The last thing in this student is very important. Question has been asked. What do they say? What is denaturation of proteins? Question was given in the paper. Many times
Yes, sir.
it's given. Again, this year can be asked question. What is denaturation of protein, students? You are saying there are different type of proteins inside your body, student, and they perform very important function. And each protein has a unique three-dimensional structure.
Yes, sir.
Each protein, student, is performing a very specific function.
Yes, sir.
And you know it has a biological activity. This is called biological activity, student.
Yes, sir.
Now this biologically active is present in the native protein. Native protein means as it is the structure which we are making.
Okay, sir.
If their structure is disturbed, student, by some method or somewhere, what will happen? When protein's structure is disturbed, then what will happen, student? Then it will lose its biological activity.
Yes, sir. Obviously.
And that is what we mean by denaturation.
Okay, sir.
Nature, D means destroying the nature of the protein. The nature of the protein is the structure of the protein. If I destroy the structure of the protein, its nature will be destroyed. And
Yes,
why
sir.
it will happen? When there is a change in temperature. If temperature is changed, students, or when it is subjected to change in pH, temperature changes, pH changes, students, then what
Yes,
will
sir.
happen? The bonds are disturbed, students. Bonds are disturbed. And if the bonds are disturbed, the globals unfold and helix get uncoiled.
Yes, sir.
What will happen then? The globules
Unfold.
unfold, student, and the helix will get uncoiled.
Yes, sir.
This is the protein will lose its biological activity. When the protein will lose biological activity, student, protein will lose the biological activity. This is called denaturation of the protein.
Okay, sir.
So this is an important question which has been asked many times. What is denaturation of the protein, students? So it will lose its biological activity, and when curd is formed from milk, student, then what happens? The formation of lactic acid by the bacteria present in the milk, the denaturation of the protein takes place. Coagulation of egg white on boiling is also example of denaturation. You know, egg, it becomes coagulated on boiling, coagulates, it coagulates, become, you have seen when you boil the egg. So these are all examples of denaturation of the protein. And a question is expected on the denaturation of protein is an important question, student. I
Yes, sir.
so this ends our proteins, and I hope you have taken note of everything, what type
Yes,
of
sir. Yes, sir.
we put.
Written everything, sir.
Okay, then good night. We will meet you in our
Okay.
next
Thank you, sir.
class. Okay, good night, students.